I'd like to pass the floor to Maja to take you into a different part of the eBrains Atlas services, which goes a bit deeper in the pipeline, into analyzing data with brain atlases. So Maja, please.
Thank you very much, Timo. I really enjoyed your presentation, so it was great. And now I will tell you about a workflow we have developed for rat and mice histological images, which is called the QUINT workflow, and enables you to do a spatial quantification of your labeled objects. So I would like to start this presentation before going to the details of the different software tools we're using to show you what you can achieve with this QUINT workflow. In this example, we had both Calbindin labeled mice sections and parvalbumin labeled mice sections. And we have performed the 3 steps. So the registration to Atlas using QUINT. Then we have performed image segmentation with the elastic tool. And at the end, we do a quantification based on the Atlas regions using the Nutil quantifier software. And those series that we used are a 6 whole brain Calbindin series, 4 whole brain parvalbumin series in mice, and we also had in this study parvalbumin series of rats. And you can find those data collections on the eBrain portals because they are released and shared, and all the tools that we are using in this study are also shared on the eBrain portal. So before digging into those tools, I also wanted to point out that on the eBrains portal, you can search for datasets. Here you can write keywords, or you can do faceted search. Once you found a dataset of interest, you click on it, select it, and you will have access to a dataset card, where you will see the name of the authors, the licenses, and the information about the data. You will also be able to visualize the data directly in the viewer, showing you both the sections and the Atlas overlays. All the tools on the eBrains portal are found under the Brain Atlas Access Services, and here for example, you can go and select the QUINT workflow. This will bring you to this collaboratory page where you have extensive explanations about the different steps and illustration. You will find information about software download, installation prerequisites, detailed tutorials and demo videos, and also address to support via the HLST contact, email, or you can also use our GitHub project to contact us. And of course, you have a lot of examples of use. So now, if we go back to the QUINT workflow, as I said, there are 3 main steps. In the first step, the purpose is to anchor your section images to the atlas. So you will use the QUINT tool to first do an affine registration, and then you can refine that using the Visualign tool. Secondly, you can use Elastic software to do a segmentation of your object, and I will have a short demo of all those tools later in this presentation. And then at the end, you use the Nutil quantifier software in order to do region-based analysis. And here you can choose between the default regions. So you will see all the regions that are available on the different atlas. Here it is in the Allen mouse atlas, but you also have the possibility to customize your own atlas regions if you're interested only in one specific part of the brain. And then when you have analyzed your results, you will get files allowing you to do 3D object visualization. So here you can see distribution in 3D viewer of the carbidin neurons and the parvalbumin interneurons. And you also get reports allowing you to do comparison. For example, here we looked at parvalbumin neuron distribution in different brain regions, both in rat and mice. So you see that there are some differences. And then also if you do look at the neuron density and compare rat and mice, you will see that in some regions there are more neurons actually in mice rather than in rats. And in other regions, it's quite similar. So now I will start with the first step, which is the anchoring of the brain section to reference atlas with QUINT. So you might wonder when you have this histological section, where is this level of the section? And if you look at 2D planches that you typically find in reference books, you might see here that some of the brain regions or the landmarks, like symbolized by those stars here, the green and the black star, they correspond to one atlas planche. But the other landmarks here in the hippocampus, they correspond to the second one. So it seems that we are in between two different planches. And why is that? This is because when this brain was cut, there was a deviation in the dorsoventral angle of 10 degrees and a deviation of 3 degrees in the mediolateral section plane. What the QUINT tool allows you to do is to tilt this 3D atlas to obtain an exact match of the atlas planche to your experimental image. And this is how the QUINT user interface is looking. So you have a main window, and when you load your section images, you will be able to see your histological images, and you have a transparency slider allowing you to overlay the atlas and look at it and toggle between the atlas and the section image. Depending on the atlas, you will have different modes. So the MRI imaging is the one that was used to make for example, here the Waxholm atlas. You also have access to some DTI information about fiber orientations. And then you have the delineations of the atlas showing you the different regions. Then you also have access to 3 small windows, showing you the sagittal, the coronal, and the horizontal planes, and allowing you to know where you are while you're working with this registration. Typically, when you open QUINT, you will get the second window where you will see the list of your sections. There is a readout of the anterior posterior level of those sections, and also an indication of if the section is anchored or not yet. I will come back to that in the demonstration. And then you have some graphical displays to show you if there are deviations in your registration. one part then of the work would be to define those angles that deviate from the orthogonal sectioning planes and apply them to all the sections. As the QUINT registration is only deforming the atlas in a linear manner, you will see that some brain regions or some landmarks are fitting your section very well. However, on the borders, it happens that it's not really exactly matching the sections. So for that, we have a second tool called Visualign that performs in-plane nonlinear transformation by allowing you to place markers into the image and stretch the 3D atlas to make it fit better. I will now go through the second step, which is image segmentation. Here we use the Elastic software. It is based on supervised random forest learning algorithm, meaning that when you have your images in Elastic, you train a classifier file by determining what is background and what is label cells, and reiterate the procedure a few times. And then once you have a classifier that is properly trained, you can apply it in batch to the rest of your images and obtain such binary images of your objects. This is done typically using pixel classification workflow in Elastic, where your objects are classified depending on intensity, color, and texture. You can also add object classification workflow if you want to refine that, and there the pixel features that are used are size and shape. So the object classification workflow is very useful to remove artifacts, speckles, or borders that typically maybe are difficult to differentiate from the labeling using only the pixel classification workflow. And then finally, we have the analysis with the util quantifier, which uses the results from the two other tools. And the graphical user interface looks like this. You will start a new job pressing new, then choose the analysis type. You'll have to define the position of the folders containing both the atlas maps and the folder containing the segmentation files. You will select the position of your coordinate file that you obtain from QUINT or Visualign. And then create a new folder for the output. You will select the color of your segmented objects. Of course, you have to select the proper reference atlas, and then save those parameters and run util quantifier, which is usually a quite quick procedure. What you will get is results in 3 different maps. So you have coordinates, images, and reports. The coordinates will give you a series of JSON files, JSON files for each of the sections, and also one combined 3D JSON. Those can be used in a 3D viewer. You can plot the objects, either one section or all the sections, like I did here. Then you get also in the second folder the images. So those are the atlas maps that are customized to your section, and then with the segmented objects are overlaid here. So you get an impression of position of your segmented objects. And then, of course, you have reports with all the quantification of your segmented objects. Now I will switch to the demo to show you quickening. So just give me a second so I can switch here. Hopefully it will work. So when you are in quickening, and I have to stop the annotation, you double click on one section. It will open Quickening software, and you will access this view of both your section and the atlas overlay. So this one I anchored already, but if I go to one section that is not anchored, so typically it would look like this. And here you have to find the approximate position in the brain, because here you see the atlas is located more anteriorly than the section. So you can do this by using the arrows or by grabbing the red pointer and move it to a part of the brain that is more corresponding to the level, and then press this double arrow, and by pressing space, it will appear a double arrow here. Place your mouse away from it, click and drag to resize and deform the atlas. So by doing this a few times, you will be able to roughly have the same size, align it. You can move in different direction using the arrows. You can also, in the main window, tilt the section like this. So here you found approximate level, and then you see that this section is not anchored because it has a red question mark. Then click store, then you get a green exclamation mark. If you go to the next section, you will see now, because I have two sections that are anchored, that even the ones that are not anchored are roughly what they should be. This one needs to be resized. Sorry. Like this. Yeah, and then here you can change, you can look at the NISL in transparency. This is useful when you have well-defined cell layers. And you can switch to the average also, sometimes it's better. And then you store. The next step is to define the deviation angles, and in order to do that, typically use a section where you have visible landmarks. So here, if I choose the hippocampus, I see that there is a small asymmetry between the left and the right side. This is because I have a small tilt that I can correct here. So here, 2 degrees tilt. And then the same thing here. I can adjust the tilt so it matches exactly my experimental image, and then store. So once you've determined that and you've saved all the images, it can look like this. And then you have all the images that have the same angles, and they are aligned to the atlas. Then the next step you can try is to go to Visualign to do a nonlinear illustration. try to switch on that. Just let me stop share for a moment. Go there and share again. So here I am in Visualine. I uploaded the JSON file I have got for QUINT, and you can see that some parts of the brain is not really matching. So here I can place a marker by pressing space, and I can drag the atlas to the correct position. So here what I'm doing is to perform a non-linear transformation of the atlas. But of course, you have to be careful because you may induce some errors that are not supposed to be here. But when you really know the anatomy and know that this border should be here, you can use that to get a better fit. And here, if you can go to that view, and it will show you all the borders. Sometimes they are easy to define, and sometimes you have no landmarks to really say what you have here. But usually by placing 15 to 20 markers, you will get a pretty good fit to your atlas map. And this result can be shared to the next tool. For Elastic, you start with pixel classification, and then you save a new project that you give a new name, or I can use the one that I've prepared already. And here, so the input data are all different images. Here we have the list, and then I can select features. So you can, there are different parameters. Usually I select everything, but you can select only a few ones if you want. There are more explanations about it on the Elastic software, and then you can proceed to the training. So here, if you label this one background and this one cell, and then you can switch. So here you will have a pencil, and so what you're interested in is showing like segmenting those black cells. So then you can mark background here, and then cells, you can mark those here. Then you can zoom in, and of course, to select the pointer, and then you can change location and zoom in more, and then label more cells. Then when you press live update, the classifier will start doing predictions of what it thinks is background and what is a cell, and then it will apply it to the whole section. So now it will take some time, but usually it's working quite well. And then, yeah, you have to wait until it's finished. And then you can export the predictions and do the batch processing. And lastly, I would like to share Nutil. So Nutil is looking like this. So here you start the operation by pressing New. You choose quantifier, OK. And then, as I showed you on the PowerPoint presentation, you have to define different folders where you have files, and on the Help button, you will have text showing you, explaining what you're supposed to do and what are the formats needed here. So you can see for the atlases, you have access to all those the versions and also the citation information. So now I will come back to my presentation and show you some examples of the QUINT workflow. I'm just checking the time. I think, yeah, I'm almost finished. So, for example, what we did here, we used sagittal mouse brain sections labeled for parvalbumin that were cut sagittally and combined those with mouse brain sections of parvalbumin that were cut coronally. And by doing this QUINT workflow registration, we could analyze the whole brain. So this is very convenient because if you do experiments, let's say in one region, and then you find some other data set on the eBrains portal that have done experiments in another modality or in another brain region, you can still fetch those images, apply the QUINT workflow, and combine both your results and results from others, and extract quantification of cell distributions, for example. The QUINT workflow can also be used for connectivity data, so it's not only for segmenting cells. Here, what we do is to, again, in Elastic, train the classifier to determine what you want. So for connectivity data, maybe we use different things. So for the injection site, which is usually a quite densely labeled region, we will, I can show you here. So here is the injection site of the fluorescent tracer. It will give a very strong signal that is seen here in red. You can use one classifier, but then for the tiny signals of the fibers that are labeled, that are connecting from one region to another, you can also use another classifier, and this is shown here in black. So you can see that we've done different analysis. I hope with this lecture, I convinced you that the QUINT workflow is very useful and will allow you to do a lot of quantifications of your mouse and rat data. We are also preparing monkey versions, but of course it depends on the size of the atlases. And for the human atlas registration, we have the Voluba tool, which is much more convenient, because then you are doing registration from 3D files, which is it's more convenient and more adapted to the human atlas. Please try the QUINT workflow and get involved in the community. You can contact us via the eBrains support address. And we have also different GitHub repositories. You can post issues, ask questions. And we also have some courses at the University of Oslo that we actually run yesterday and today. to learn how to use those tools. And lastly, I would like to thank all my collaborators in the Ursprünglich Forschungszentrum, the MBL in Heidelberg, and the HPP. Thank you for your attention, and please tell me if you have some questions.